Okay, I'm going to start with this animation of the second part of, I think you could say that we usually call it the second part of gene expression, or the central dogma of biology, which is called translation. And translation is how proteins are made using the information that's in the mRNA, and I'll explain what I mean by that, for the ribosome to use that information to make proteins, different kinds of proteins. And what makes different kinds of proteins is really dictated by the order in which the amino acids are linked together. Because if you link together amino acids in certain orders, they will fold into different specific shapes, and that will dictate different functions. And so when we talk about different proteins, we're talking about different primary sequences of amino acids in the polypeptide chain, which determines the three-dimensional shape and therefore the function of that particular protein. Okay, so we're going to watch this real quick, and then I think we'll watch it again, honestly, when we are at the end of this section.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so it's a good start because this process is more complicated than transcription. So let's start with, in translation, there is something important called the genetic code. And make a note of this. The genetic code has nothing to do with transcription. Transcription, the RNA polymerase just copies whatever it sees in the gene. Whatever the nucleotide sequences it copies it. But in that mRNA, if it's an mRNA, there are something called codons. But transcription doesn't pay attention to codons. Translation pays attention to the codons. Now, what are codons? It turns out that some scientists you may recognize, Francis Crick, yes, it's the same Crick from Watson and Crick who discovered the structure of DNA. And another scientist, Sidney Brenner, has won the Nobel Prize twice. And he was also working on trying to figure out the structure of DNA, although he didn't get it. But they went on to figure out that there had to be some kind of information in the chromosomes, in the genes, that somehow that information got transmitted out of the nucleus to the ribosomes, which are in the cytosol. And so they speculated that there would have to be a messenger molecule, and it turns out there is. It's a molecule of RNA that copies the information from the gene in the nucleus, and then the mRNA gets modified, and then it comes out and it finds the ribosome, and the ribosome clamps down on that mRNA, and as you saw in the animation, there's a whole process then by which the information in the mRNA causes the construction of the proper protein, the amino acids in the correct order. And so what they figured out was that in the DNA and in the mRNA, there are groups of 3 nucleotides, one right after the other, and these are called codons. And each group of 3 nucleotides corresponds to a particular amino acid type. And so there's the tRNA molecule is the kind of the go-between that connects the correct amino acid and brings it into the ribosome with the correct codon. And so there were some other scientists, Nuremberg and Korana, who figured out which codons, which 3 nucleotides in the mRNA indicate which amino acid to be brought in by the tRNA. And I'll show you the code. But what's important about the code, first of all, is that there are 3 codons that are called stop codons. Stop codons do not have any corresponding tRNA. So when a stop codon comes into a ribosome, that's what happened at the very end of the animation when everything stopped. But there's also a start codon that tells the ribosome where to start on the mRNA. And there's only one start codon, and that codon is AUG. So all genes start with AUG in the mRNA, BATG in the DNA, and that would signify the start of translation. And AUG actually does correspond to an amino acid called methionine. So all proteins, when they're first made, start with the amino acid methionine because all genes start with the codon AUG. So this is the genetic code, at least the way it's often shown. And what you're seeing here is the 3 nucleotides. Get my pen here. The 3 nucleotides, and then which amino acid it corresponds to. So UUU in the mRNA will cause the ribosome to get a phenylalanine from the tRNA. UUC will also cause a phenylalanine to be brought in, whereas UUA causes a leucine to be brought in, and same with UUG. So typically the amino acid names, these are the amino acid names. Most of them end in INE, but you do have a few that don't. Let's see, tryptophan and aspartate and glutamate. So a couple that don't, but most of them end in INE. And in the amino acid language, we use either a 3-letter abbreviation. You don't need to memorize it. I will provide this code for you if you need it for answering a question on an exam, for example. But these are the 3-letter abbreviations and then the full name of the amino acid. So these codons in the mRNA would correspond to the ribosome putting a leucine into the protein. These codons in the mRNA would cause the ribosome to put a proline into the protein. So as the ribosome basically is bringing the mRNA through it, the corresponding tRNA is bringing the correct amino acid that corresponds to the codon in the mRNA. Now, if there's a mistake in the mRNA, there's no way the ribosome could know, and there's no way the tRNA could know. Each process is, doing one job. And so if there's a mutation in the DNA, which gets copied into the mRNA, then you could get the wrong, with a codon that wasn't supposed to be there coming into the ribosome. But that could mean that you get the wrong amino acid put into that protein. So that does happen. But assuming everything is normal, everything is correct, then every time that same mRNA is translated, the same protein will be made. The same amino acids will be hooked together by dehydration synthesis. Right? Okay. Oh, and one thing I want to point out. So you notice there's more than one codon for each amino acid. The only exception to that is methionine. There's only one codon for methionine, and all proteins start with methionine. So that's where translation begins, at the start codon with the incorporation of methionine as the first amino acid. So the genetic code is said to be universal, but it turns out it's not completely universal. It's practically universal, meaning almost all living organisms have the same code, the one I just showed you, except there's a few minor differences. Like in mitochondria and chloroplasts, sometimes there are slight different stop codons. There's some like one-off organisms that might have one codon that's different. But for the most part, very, very high level of universality. And so what that means is that you can take genes from one organism, the genes, the DNA, the chromosome, put it into the cells of another organism, and that organism can still perform transcription and translation and make that protein in that other organism. So here you have a transgenic pig, the one that has the yellow snout. They've taken a gene from jellyfish that creates this kind of glowing color here, and they have inserted it into the chromosome of a pig. And even though it's a jellyfish gene, the pig can make the jellyfish protein. And so you can see that that protein appearance is this yellow kind of glowing color. So because that's true, because the same genetic code and the same process of transcription and translation happens, you can transfer genes between organisms at the DNA level, and the cell can still perform those processes of transcription and translation on the genes from other organisms. And so that creates all kinds of possibilities in biotechnology, the ability to transfer genes from one organism to another. And I will give you a really good example of this. One of the first genes that was kind of at the beginning of the biotech era, they were able to take the gene, the human gene for insulin, and they put it into bacterial cells, and the bacteria produce human insulin by transcription and translation. And that's where our supply of insulin for diabetics comes from now, is from bacteria that are making human insulin from the human gene that's been inserted into those cells. So this has made all kinds of things possible, right? So it's important that it turned out to be universal. If every organism had a different code, then you would never be able to transfer genes between organisms and expect to get the same protein produced in them. Okay, let's talk a little bit about the tRNAs. tRNA is made by transcription, so there are genes that encode the tRNAs. And then once the tRNAs are produced, it's just a polynucleotide chain. Where's my, get my pointer. And so when the gene is transcribed, you get a polynucleotide chain that folds into this clover leaf shape. So see, this is called the clover leaf. But in reality, it's not flat like that. It's more three-dimensional. So it might look more like this. This is a space filling model, and this is what the artist in your textbook is going to use to draw when it's drawing a tRNA. So what they're focusing on is at one end of the tRNA is where the amino acid attaches, the acceptor end, and at the other end is this very special 3 nucleotides called the anticodon. The anticodon is complementary to the codon. So, for example, if the codon is AUG, then the tRNA that carries methionine, which is the start amino acid, the anticodon tRNA of the tRNA would be UAC. And because those are attracted together, that's why that tRNA is attracted into the ribosome when that AUG codon is of the mRNA is in that correct spot. So it draws that tRNA into the ribosome by that attraction. And then basically the tRNA drops off that ribosome and leaves, if you saw it in the video. So the anticodon is complementary to the codon in the mRNA. And so each tRNA has a different anticodon and carries a different amino acid. All right. But the genetic code that I showed you is not written in anticodons. It's written in the codons that are in the mRNA. So the fact that you have these tRNAs involved in the process, that's not reflected in the genetic code. You just know it because you know how translation happens. Now the ribosome, we learned about the ribosome in chapter 4. Its job is to, move along that mRNA and allow the correct tRNAs to come in and then make the peptide bonds between the amino acids. So that's the dehydration synthesis that joins the amino acids together. The enzyme that performs this, which is an enzyme that's built into the structure of the ribosome itself, is called peptidyl transferase. So that's where the dehydration synthesis of these incoming amino acids happens because of that enzyme, which is part of the ribosome. So this picture is really taken from, this is a micrograph or a really good resolution space filling model. I can't even tell. It's a very good image of a ribosome. This is more of just an artist drawing. So a lot of times when you see an artist drawing of a ribosome, it looks like this. It's actually in 2 pieces. You have, and to me it looks like a hamburger bun. So, sorry, it looks like a hamburger bun to me. You got the top and you got the lid of the bun and you've got the bottom of the bun. So to me it looks like a hamburger bun. Do not write that on the test. But we call this the large subunit and the small subunit. And in between the large and the small is where the mRNA goes. So the mRNA goes between the large subunit and the small subunit. So I'm telling you, it's a bacon sandwich where the mRNA is the bacon. Now, if you're a vegetarian, I'll have to come up with a different analogy. Okay, but the mRNA gets sandwiched between the large and small subunit. It's a good way to think of it. And the mRNA, one thing to know, even though in the video it was a little misleading, the start codon is not all the way at the very tip of the mRNA. The start codon will be a little bit down from that 5' end. And the stop codon is not all the way at the very 3' of the mRNA. It'll be a little bit up. So in reality, let me draw this. If this was an example, you would have like maybe the start codon is like right here, and maybe the stop codon is right here. You know what I'm saying? So it's not, and so the ribosome will start, it'll kind of scan along and it'll start here and it will stop here. So there's always this little bit of extra on both ends that doesn't have codons. And that's okay. That's the way it always is. So the ribosome has to scan along, and it finds wherever there's an AUG, and that's where it starts. So it's close to the 5' end of the mRNA, but not at the very 5' end. And then once it starts, it's going to bring in this diagram, I guess, this is amino acid 1 has to be methionine. So let me put MET first. MET. So methionine would come in first because the codon is AUG right here in the mRNA, and the tRNA would be UAC, which is attracted to that start codon. And so that tRNA that has a UAC anticodon always carries methionine. And so it's attracted in, and then it drops off the methionine, which is just this green ball in this diagram. And then that tRNA is going to then leave the ribosome. So the transfer RNA is literally going out, picking up the different amino acids. Each tRNA with its own particular anticodon picks up a particular amino acid. And then when that codon comes into the ribosome, that tRNA is attracted in. And so one by one in order, the ribosome is building the amino acid chain according to the codon instructions of the mRNA. And at the end of the mRNA, there will be a stop codon. So if you have 100 codons in the mRNA, you would only get 99 amino acids in that protein because each codon signals for one amino acid, except the last codon does not signal for an amino acid. It signals for the process to stop. All right, so that's what I mentioned in the next slide. Termination of translation. That just means how does it stop? When the stop codon comes in, so here's an example. One of the stop codons is UAA. There is no tRNA that has an anticodon for that. And so what you're seeing here is that protein is then released, and these two subunits of the ribosome come apart. Dissociation just means release or come apart. And then that protein will then fold into its three-dimensional shape. And one thing that's nice about this is this mRNA can be reused over and over again to keep making that same protein, many of those same proteins, right? Because it still has all the information in there. However, RNA in the cytoplasm is a little bit short-lived, especially mRNA. So the mRNA won't stay around forever. Eventually it will be degraded or destroyed. So ultimately, if a cell wants to continue making a particular protein, it has to keep doing transcription, so it keeps sending mRNAs out to the ribosomes to keep that process going. You can't just make one mRNA and then expect it to last forever. It's not going to. Okay, so let's watch this again. I think it is helpful.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.